Welcome to Watson and Crick DNA, explained by my good friend Ollie the Owl. We will begin by exploring the basic structure of DNA. The basic structure of DNA is made up of three major components: a nitrogenous base that's either adenine, cytosine, guanine, or thymine, a phosphate group, and a deoxyribose 5-carbon sugar. Adenine and guanine are both purines because they have 2 rings, while cytosine and thymine are both pyrimidines because they only have 1 ring. Now let's talk about base pairing. Base pairing is how the 4 nitrogenous bases pair together with 1 purine and 1 pyrimidine. The complementary pairs are adenine and thymine and guanine and cytosine. I've highlighted the A's and T's in blue and the G's and C's in green. The sugars in DNA are named after their functional groups. Carbon 1 is where the sugar attaches to the nitrogenous base. Carbon 2 contains 2 hydrogens, but in RNA it has another hydroxyl group. Carbon 3 contains the hydroxyl group, which is significant because DNA builds off of the free 3' end. Carbon 5 is attached to the phosphate group that is also attached to the next nucleotide's 3' carbon. This pattern continues up the whole length of the DNA molecule on both strands. DNA is also anti-parallel, meaning that each strand runs in the opposite direction, as shown by the 5' and 3' ends. Being anti-parallel allows the complementary bases to pair, and also allows the formation of hydrogen bonds down the middle of the molecule, which makes it more stable. The stacking of these bases, along with the arrangement of the sugar-phosphate backbone, make up the basic structure of DNA. The major groove of DNA occurs where the backbones are far apart, and the minor groove occurs where they are close together. Now let's look at handedness of DNA molecules. Starting with the one on the left, if Ollie the Owl goes to walk up this staircase, he will need to put his left hand on the railing in order to walk up it, making this a left-handed double helix. Now if Ollie tries to walk up the staircase on the right, he will need to put his right hand on the railing to go up the staircase, making this a right-handed double helix. Tm is the melting temperature. Let's take a look at what would happen if we placed our DNA molecules in a high concentration of salt solution. So let me pour the salt solution all over our DNA molecules. The positively charged sodium in the solution will go and find the negatively charged oxygen in the phosphate groups. Sodium is the cation of the salt, and the positive charges neutralize the negative charges on the DNA phosphate backbone, eliminating the repulsive forces there. This increases the Tm of the DNA. Additionally, the salt makes the DNA molecule less hydrophilic, which makes it less soluble in water.